reactions that are happening inside of a cell. And remember that you've got two types. You've got catabolism and anabolism. Catabolism is when you do what? Break stuff down. Okay. Anabolism is when you put it together, build stuff up. So your cell is constantly breaking down nutrients, carbohydrates, lipids, not so much proteins, although it can if it has to, right? But our cells prefer to use carbohydrates first, and if we run out of carbs, they use lipids, and if they run out of lipids, you're screwed, because you basically start digesting yourself. So these organic molecules, these nutrients, are transported. They either diffuse or actively transported. We know all about that now, right? Inside the cell, and they're basically hanging out of the cytoplasm, and that's part of the nutrient pool. And then some of those nutrients are used, they're broken down in the cytoplasm and in the mitochondria to make ATP. So some of those nutrients are catabolized to produce energy. And then that energy and other of the nutrients are used anabolically to build proteins that the cell needs. The cell needs to make a certain kind of receptor or a certain kind of enzyme, or needs to make a vesicle, needs to make a lysosome. So anything the cell needs to make, anything the cell needs to anabolize, you have to have the raw materials. That's what some of those nutrients are used for, as well as energy. Other nutrients are used to make that energy. So there's this whole cycle of catabolism and anabolism that's going on inside of a cell. Now, when your cells or when your digestive system, I should say, before this stuff even gets into your cells, in your digestive system, when you eat food, okay, if you eat carbohydrates, those are digested or broken down into monosaccharides. So, glycogen, starch, those are polysaccharides, right? So we have to break those down. Our digestive system, again, this is in the lumen of your digestive system before anything even gets into the cell. Lipids, triglycerides, are broken down into the glycerol and the 3 fatty acids. And then, of course, proteins are broken down into what? Amino acids. So it's the job of your digestive system to get your food into small enough molecules that can be transported into your cells. So, once these things, monosaccharides, glycerol, fatty acids, amino acids, once those things are transported across the cell membrane into the cell, amino acids, monosaccharides, fatty acids, glycerol, those things are actually, by themselves, are too big to get into the mitochondria. So they've got to be broken down even further before they will go into the mitochondria. If you take glucose is C6H12O6, right? It's got to be broken in half into 2 3-carbon molecules. Glucose, that monosaccharide, is too big. It's got to be broken in half. That has to happen out here in the cytosol. Fatty acids. You could have an 18-carbon fatty acid. That's way too big to fit into a mitochondria. So fatty acids are broken down 2 carbons at a time. And those 2-carbon fragments are small enough to enter the mitochondria. So your digestive system has to break the food down into molecules small enough to get into the cell. And then enzymes in the cytoplasm, in the cytosol, have to break them down even further to get into the mitochondria. Your cells prefer to use carbohydrates for energy because they tend to use lipids and proteins for anabolism. So we're going to talk about glucose catabolism. We're going to talk about breaking down glucose to get energy. All right, once a glucose molecule is transported into the cell, there are basically 4 things that have to happen in order to extract the energy, to get energy out of that molecule of glucose in order to make ATP. All right, the first step is glycolysis. Now, if you look at the word glycolysis. Lysis means to break it down. Yeah, rupture, blow up, break apart, right? Glycolysis happens in the cytosol of the cell. Glucose is broken apart in a series of steps. One glucose molecule is basically broken in half to form 2 pyruvate molecules, or 2 pyruvic acid molecules. So glycolysis is the process of taking glucose and going through a whole bunch of steps to form 2 pyruvate molecules. Pyruvic acid, same thing. If you have it in a jar, it's pyruvic acid. If you put it in water, it's pyruvate. And that occurs out here in the cytosol of the cell. Pyruvic acid, the 2 pyruvate molecules, get transported into the mitochondrial matrix, they get converted to something called acetyl-CoA. That happens in the matrix of the mitochondria. Then the acetyl-CoAs enter what's called the citric acid cycle. And the citric acid cycle produces a bunch of electron carriers. And those electron carriers carry the electrons over to the inner membrane, to the electron transport system. The electron transport system is a bunch of little molecules. It's actually embedded in the inner mitochondrial membrane. And it's finally at the electron transport system that you get most of the ATP. If you'll notice, the only part of this that doesn't require oxygen is the first step. The only part that doesn't need oxygen is glycolysis. You know when you say you do aerobic exercise? Okay. What you're doing, when you're doing aerobic exercise, that means your cell's doing all of this. Because it has to do this before it can do this, right? Okay. But you know when you push your muscle cells too far, and you're going to that maximum amount of effort, and you say you're now in anaerobic metabolism? All your cells are able to do. There's not enough oxygen going to your cells to do the rest of this, so all they're doing is this part. So all they're doing is they're taking glucose and they're breaking it down to pyruvate, and that's all they can do. There's not enough oxygen for them to do the rest of it. In fact, what happens if not enough oxygen is getting to the cells, the pyruvate never gets transported into the mitochondria. What happens is the pyruvate gets converted to lactic acid, the pyruvate to acetyl-CoA in the citric acid cycle. This doesn't directly use the oxygen. In fact, the oxygen is not directly used until the very end of the electron transport chain. But if there's not enough oxygen diffusing into the cell, these two things don't happen. So they're considered part of aerobic metabolism, even though the oxygen is actually not used directly until the end of the electron transport chain. All right, so now what we're going to do is I'm going to start a little bit talking about glycolysis. We'll talk about the rest of it next week. All right, I said that in glycolysis, glucose, which is a 6-carbon molecule, right? C6H12O6? Okay. Yeah. Glucose is a 6-carbon molecule. It gets broken down into 2 3-carbon molecules. That's what pyruvate is. One big 6-carbon molecule broken basically in half into 2 3-carbon molecules. And of course, they're small enough to get into the mitochondria. And that's the whole purpose of doing that, right? Now, this is happening in the cytosol. It doesn't require oxygen. And there's a whole bunch of enzymes involved that we don't have to know about. Every place you see one of these arrows is a different enzyme, but we don't have to know them. The way this works, you know how you have to spend money to make money? So that's what the cell has to do. The cell actually has to use 2 ATPs to get this started. So it actually has to use a little bit of energy. But what happens at the end, you get 4 ATP back. So once you finally get down to the 2 pyruvate molecules, you get a total of 4 back. You spent 2, you got back 4, so you profited 2. Here's what you need to know about glycolysis. This is the bottom line. When one 6-carbon molecule of glucose gets broken down into 2 3-carbon molecules, you get out of that 2 ATPs and 2 molecules called NADH. NADH is an electron carrier. Now, why is electron carrier important? Because you're going to carry those electrons over to the electron transport chain and make a bunch of ATP. So we did glycolysis, right? Got our pyruvate. Okay. The pyruvate, which each of those has 3 carbons, basically a carbon is ripped off of it. And the pyruvates get converted to acetyl-CoA. So you get 2 pyruvates. They're converted to 2 acetyl-CoA molecules. Each of those having 2 carbons. So basically, when the pyruvate molecules enter the mitochondria, the first thing that happens is they get converted to this molecule, and each one gets converted to acetyl-CoA, which only has 2 carbons. That 3rd carbon is basically converted to carbon dioxide, and you breathe it out. Because when we break down glucose, we use oxygen and we produce carbon dioxide, right? That's where the carbon dioxide comes from. The carbons that were originally in the glucose. Eventually, we're going to break it all the way down into just carbon dioxide. Now, again, what you need to know about this, the bottom line is this. We get an additional 2 NADHs. The energy, the stuff we're going to use to make ATP, it's basically stored in these electron carrier molecules. So we took glucose, which had 6 carbons. We broke it in half, made 2 pyruvate molecules. When we did that, we got out of that 2 ATP, right? And 2 molecules of NADH. Now, ATP is ATP, so we can use that directly, right? Okay. But there's still some energy. These things are carrying electrons, right? So we're going to use this to make more ATP later. Okay? Then the pyruvates entered the mitochondrion. Each one of the pyruvates was converted to acetyl-CoA. Basically, our carbon was ripped off, converted to CO2. From that process, we get an additional 2 NADH. Then the acetyl-CoA enters the citric acid cycle. Now, depending on what textbook you read, in the olden days, it was called the Kreb cycle. Citric acid cycle, Kreb cycle, or TCA cycle. It's the same frickin' cycle. I will probably slip up and call it the Kreb cycle because that's how I learned it. Here's the important thing. It does occur in the mitochondrial matrix. Oxygen, it is aerobic because it won't happen unless oxygen is around. Per acetyl-CoA that goes into the Kreb cycle. Now, how many acetyl-CoAs do we have in total? 2. But for each acetyl-CoA, out of the Kreb cycle, you get 1 GTP, which is kind of like a generic ATP, 3 NADHs. We get a second type of electron carrier called an FADH2. The acetyl-CoA had 2 carbons in it, and that's where the other 2 carbon dioxides come from. But we have 2 acetyl-CoAs in total. So in total, we end up with how many of these?
2.
How many of these? And how many of those?
2.